very much for giving me this opportunity to talk to you about some of the technologies that we're developing in Queen's University Belfast. I'm going to talk to you today about DNA vaccination. I promise to stay within the time limit. We're going to use the RALA peptide to deliver our genes inside dissolving microneedles. Okay, the prototype that we're using it for, so proof of principle, the model that we've selected is for cervical cancer. And again, as Paul Fisher this morning explained, cervical cancer is the second leading cause of death in cancers for women. HPV is present in about 70 to 99% of all cervical cancers. HPV 16 and 18, these are the key ones. These account for 80% of all cervical cancers. And they produce the oncoproteins E6, E7. And the significance of this is that the oncoproteins E6, E7, E6 binds to the P53 tumour suppressor gene and prevents it from doing its role. And the E7 binds to the retinoblastoma, PRB, protein, again preventing that. It is in control of cellular proliferation. So whenever they bind to both of these proteins, there's uncontrolled cell growth. So, the current vaccination strategies within the UK, Gardasil is quadrivalent vaccine, and it contains 4 different serotypes, 16, 18, 6 and 11. And Cevarex, which was approved in 2009, is a bivalent vaccine containing 16, 18. The problem is it's a prime boost regimen. It's over 6 months. And it's only prophylactic, okay? So it's not going to prevent, it's not going to have a therapeutic response if you've actually got cervical cancer. The other big problem we have in the UK is we only have around a 38% compliance. So it's not very good. So why DNA vaccine? Well, Paul touched on this, because we can produce both cellular and humoral responses, and it's really the cellular responses that we're looking to get. If we can get target antigen presenting cells, they will present on the MHC class 1, and what will then happen is you'll get activation of the CD8 cells. If you get activation of your CD8 cells, your CD8 killer T cells, that will result in destruction of the actual disease. So in that way, it can be therapeutic as well as prophylactic, where you get the more traditional CD4 response. It's reproducible and cost effective. You can manipulate your plasmids very effectively. It's very safe, no chance of vaccine reverting to virulence, and more importantly, it's very stable, so you can transport it around the world, no need for cold chain storage. However, as many people have discussed, the rate limiting factor upon this becoming a wide stream therapeutic is the delivery issues. So basically, you've got issues in the extracellular environment here. Binding and aggregation are an issue. Once you get extravasate out of the capillaries, you've got to get across the cell membrane. Once you get out of the cell membrane, you've got to get out of the endosome. And then, in order for the DNA to be transcribed and translated, it has to get into the nucleus. And unless you have an active nuclear transport system, it will only get into the nucleus on a chance effect, because during replication, the nuclear membrane dissolves, and that's when it will be able to get in. So, a lot of issues to overcome. Our solution in Queen's is twofold. In order to overcome the extracellular barriers, we use the strategy of microneedles, and there was a very good talk explaining microneedles yesterday. And we use polymeric dissolving microneedles. These are only 600 microns in length. In a 1 by 1 centimetre patch, we get about 361 projections. And these are designed to penetrate the stratum corneum, not go deep enough to cause any pain, and dissolve and release the cargo. And in order to overcome the intracellular barriers, we have designed within my research group the RALA peptide. It's a 30 amino acid peptide. It's designed to protect DNA. It condenses DNA into very small nanoparticles. It will disrupt the endosomes and aid nuclear localization. And that's just a typical picture of our nanoparticles that we get with DNA. And indeed, we published all about the RALA peptide in JCR in 2014. So, we just picked a plasmid that coded for E6, E7. And the nanoparticle formulation was essentially the E6, E7 DNA was added to the RALA peptide, as simple as that, and just formed nanoparticles in double distilled water. And if we have a look at it in a little bit more detail, a couple of things I'm going to point out to you is that The RALA peptide has an arginine face, it has a hydrophobic leucine face. When you condense them together, it is not like your conventional nanoparticles where you would get maybe your DNA in the middle and your nanoparticle round the outside. This is more of an interaction like this, so the RALA is the whole way through the DNA. We also put in tryptophan residue. Tryptophan is particularly good. It anchors in the membrane of the cell. And one of the reasons why arginine is such a good thing for any cell-penetrating peptide is the inner leaflet of the phospholipid bilayer has phosphatidylserine. It binds with higher affinity to arginine, and it actually forms a membrane capacitance effect. So it pulls a nanoparticle through the cells. Once it gets through the cells, and we've shown this is mainly by clathrin-mediated endocytosis, it's taken into the endosome. At that point, there's a drop in pH, as we know, and the peptide becomes more alpha helical. We've shown this with circular dichroism, and that alpha helicity then enables it to escape from the endosome into the cytosol. Okay, so we made our nanoparticles. We wanted to check the functionality of it, and so what we could see is that from NP ratio 2 upwards, we get relatively small nanoparticles. They're less than 100 nanometers, give or take 1 or 2. And as you would expect, cationic, positively charged. NP ratio was just the ratio of the nitrogenous atoms in your peptide to the phosphate in the backbone of the DNA. Most importantly, though, RALA is less cytotoxic. So in fibroblast cells, NCTCs, we examined the cytotoxicity and looked at surviving fraction. Compared to lipofectamine and polyethylamine, this was a lot less toxic. So that's the nanoparticles. Let's manufacture the microneedles. It's a two-step manufacture process. So the nanoparticles are placed in the microneedles only. Okay. So the nanoparticle loaded polymer matrix is put into these kind of silicon molds that we've had lasered out, and then it's centrifuged down. We used 20% polyvinyl pyridine. We centrifuged them down, and then we added an empty polymer for the base plate. We then allowed them to dry for 48 hours before removing them from the silicone molds. At that point then, we cut off the sidewalls, and then we were left with our microneedle patches with the nanoparticles in the tips, in the needle part of the microneedles. Okay, so we needed to determine what our functionality of our nanoparticles were in the PVP matrix. So we we took the PVP matrix and we dissolved it after 0 days and after 7 days. So A is 0 days, B is 7 days, using delivery of a GFP reporter gene. And what you can see is that we do have expression. There is functionality in panel B at 7 days. We then look to double confirm this, delivering E6 DNA on its own, E7 DNA on its own, and then the fusion one of both, which is the E6, E7. And what you can see from the Western blot here is after 7 days, and these are 7 days of the nanoparticles being in the PVP, we have indeed got expression. Okay, so then we went on to look at in vivo delivery in mouse ears, and for those of you interested in the microneedle field or indeed in DNA delivery, I'd be very happy to have a conversation about how you accurately measure delivery of DNA. It's a very, very difficult thing to do. But what you can see in the lower panel, for those of you that can see that, is we Cy3 labeled the DNA, and we applied the patches to the ears of mice, and we're interested in that intradermal layer again because there's a high proportion of antigen-presenting cells. And you can see very clearly there are red nanoparticles there, or sorry, red pieces of DNA there. So we were able to confirm that we had delivered the DNA to the mouse ears. Of course, we then wanted to have a look at the penetration, and we do a thing called optical coherence tomography, where we use different forces of the microneedles. So we gave 10, 16, 18, and manual force. Manual force is in around 20 newtons. We have also designed a patch where it's got a backing plate on it, and when you apply the right pressure, it turns red. But the thing that I want you to notice, I suppose, is that this is your stratum corneum layer, just here. And you can see that the microneedles have penetrated down, but most importantly, no matter what force we used, by about 15 minutes, they had completely dissolved, and the skin was beginning to reclose over again. Okay, so most importantly then, we wanted to see what the effects were. So what about our antibody-mediated immunity? So what we did was an indirect ELISA for E6, E7 specific IgG antibodies. And this is indeed a typical ELISA plate from this. Control, empty microneedles. Positive control, which was a peptide for E6, E7. DNA injected intramuscularly. DNA injected intramuscularly. DNA inside the microneedles. Note, DNA inside the microneedles didn't produce any antibody response. Nanoparticles IM, so that's just the RALA E6, E7 on its own, and the nanoparticles then inside the microneedles. And what we found is that microneedles with RALA E6, E7 nanoparticles evoked a more consistent humoral mediated response. So that's good, but what we're really looking for is a CD8 response. Okay, we really want to see the ability of this system to evoke that kind of therapeutic response. So we carried out a cytotoxicity assay to test the immune response basically of our product. So we immunized the mouse with our DNA patches and we put it on the ears of the mouse. We homogenized the mouse spleens. We co-cultured with irradiated TC1 cells. This is a cervical cancer cell that expresses the E6, E7 antigen. And when we co-cultured them, we took off the supernatant and did two assays. Okay, some of the supernatant was first of all co-cultured with viable T cells, and we detected then the level of cytotoxicity. This is what we wanted to see how much cell kill there would be, and then we took the supernatant of each sample and measured interferon gamma. For those of you that don't know, interferon gamma is a surrogate marker of CD8 response. And what we found is that both our cytotoxicity assay and also with the interferon gamma assay, that we got a really good significant response with our nanoparticles and microneedles. This is cytotoxicity assay. 5 to 1 and 10 to 1 are just the ratios of nanoparticles that we used. And that in our interferon gamma assay, the best response after the positive control was the nanoparticles in the microneedles. Of course, we really wanted to see what the effect on tumours were. So, to that end, we performed two different experiments. We first of all did a tumour prophylactic study where we took C57BL6 mice and we immunised them in a prime boost regimen with 2 weeks apart, weekly intervals. 1 week after the last vaccination, the mice were then challenged with the tumour cells and the mice were monitored for evidence of tumour growth. Alternatively, we reversed it and we first of all gave mice 1/10⁵ TC1 tumour cells on the rear dorsum, and whenever they reached 50 millimeters cubed, we then gave the vaccination regimen as per the prophylactic study. So a prime boost. We have a look at the prophylactic response first of all. You can see quite significantly that the percent survival was in fact the highest in the nanoparticles in the microneedles group. Okay, that's the red line compared to all the other groups. And indeed, out of we have 7 mice in this group, we had 4 that had complete responses. They never developed tumours in the first place. But you can see the general trend of basically improved efficacy with the nanoparticles in the microneedles. Conversely then, we looked at the therapeutic response and we saw exactly the same effect. The best survival was in those mice that had been vaccinated with the nanoparticles in the microneedles. And again, we had a few where the tumours completely disappeared. But there is a general shift, as you can see, upwards in the red again, of the nanoparticles in the microneedle group responding the best. However, when it comes to DNA vaccination, we were delivering in a patch approximately 10 micrograms. These mice were vaccinated with 60 micrograms, so that needed several applications of the patch. So we needed to work on the formulation to try to improve the dose. So to that end, we did a freeze-drying protocol where we made up the nanoparticles at an MP ratio of 6. We added 5% trehalose, not as a traditional cryoprotectant, but actually in this case so that we could see the product after we freeze-dried it, because it's very difficult to see freeze-dried nanoparticles without this. We reconstituted then in 20% polymer. We loaded it in the microneedles and we applied the microneedles on the mice ear for 24 hours, and then we quantified what was left in the patch. I agree that it's not exactly an absolute, but it gives us an indication of approximately how much DNA that we can deliver. We looked at a couple of different polymers. So the one that I've been talking about so far has been the 360 kilodalton PVP. We also looked at a 58 kilodalton PVP because we wanted a lower molecular weight so that it wouldn't affect toxicity throughout the body. And we also looked at a 9 to 10 kilodalton PVA polyvinyl alcohol. And what we found, the red at the bottom, it says before freeze drying, after freeze drying, and then after freeze drying. And in red underneath it tells you how much DNA was put into the patches. And what you can see is when 100 micrograms of DNA was loaded in the patches, that we're getting over 70 micrograms of our DNA back out. Now this is a 1 by 1 centimetre patch, so this is very significant how much DNA we can actually deliver in that one patch. That means that we can then put that patch on the ear of the mouse and we don't need to put multiple patches on for that vaccination. So it's all very well and good freeze drying all of this. We wanted to check the functionality. So we used the PVA patches and we had freeze dried and loaded 100 micrograms of DNA. And what we basically did, we looked for the therapeutic response. So we implanted the mice with tumours. Whenever they got to 50 millimetres, we did a prime boost regimen again. And after 21 days, we excised the tumours from all of the groups. I think it's pretty self-explanatory, but you can see the control and empty microneedles and the E6, E7 peptide did not evoke a therapeutic response whatsoever. The DNA IM a little bit, DNA microneedles a bit, but by far the best group was the nanoparticles in the microneedles. And again, we've just quantified that on the right-hand side with the tumour weight. So where are we at now? Well, we're examining other polymers. We don't have an optimized construct in terms of our DNA. This was just a proof of principle to see if we could do this. And we're looking at the impact of adjuvants, so we could deliver DNA for GM-CSF to encourage T-cell proliferation. And also, we could also do mixed patches, so we can do bivalent, quadrivalent in one single patch. I'd like to thank you for listening to me, and this is just some of the members of my group, and I'd also like to thank my co-investigators. That's it.